The overall goal of this procedure is to perform high-throughput screening of antibacterial effects of nanoparticles with or without conjugation to antibiotics. Here, the antibacterial effects of polymyxin B conjugated cadmium telluride quantum dots are tested. To accomplish this, the quantum dots are first solubilized in mercaptopropionic acid. Next, the quantum dots are conjugated to polymyxin B antibiotics. Plated bacteria are then treated with both antibiotic conjugated and unconjugated quantum dots, and half of the samples are subject to irradiation with 440 nanometer blue light for 30 minutes, which causes the quantum dots to generate excitons and reactive oxygen species. To assess the effect of quantum dot treatments on bacterial growth and survival, optical density readings are taken and colony counts are performed. The resulting data indicate that application of antibiotic-conjugated cadmium telluride quantum dots followed by irradiation yields the greatest bactericidal effects amongst the conditions tested.
Though this method can provide insight into the effect of cadmium telluride quantum dots and their conjugation to polymyxin B on E. coli growth, it can also be applied to other types of nanoparticles and antibiotics on various bacterial strains.
Generally, individuals new to this method will struggle because any excess of MPA will make the quantum dots aggregate, and if they pipette out toluene, even a small amount, they will destroy the filter. Although today we will demonstrate a method for assessing the antibacterial effects of antibiotic conjugated cadmium telluride quantum dots, similar method could be used with other type of quantum dots, such as indium phosphide zinc sulfide and cadmium selenide zinc sulfide.
Begin this procedure by preparing a solution of cadmium